All right, hopefully you guys can see my screen. So hi everyone, my name is Clayton Otter. I'm a third year MD PhD student at the University of Pennsylvania in the laboratory of Susan Weiss. And today I'm going to be talking to you about my poster, which was unable to happen yesterday due to technical difficulties. But it is called MERS coronavirus NS4A, NS4B, and ENDO-U antagonized double-stranded RNA-induced innate immune pathways. And this work was done in close collaboration with a previous grad student in the lab, Courtney Komar, who just graduated. So much of this data is hers as well. So I'll just get started. So MERS stands for Middle East Respiratory Syndrome. It's caused by a coronavirus that first emerged in 2012 and since then has caused over 2,500 laboratory confirmed cases, with over 900 of those resulting in deaths, giving a case fatality rate of over 36%. Recent work in the Weiss lab has focused on evasion of 3 antiviral innate immune pathways by MERS. To study these pathways, we've used the A549 cell line, which we've engineered to express DPP4, the cellular receptor for MERS. After binding to its receptor, MERS is internalized via endocytosis and releases its single-stranded RNA genome into the cytoplasm. All coronaviruses produce double-stranded RNA as a byproduct of their replication, which is recognized by various cellular immune sensors, 3 of which we've been particularly interested in. The first of these is MDA5, which after recognizing double-stranded RNA induces an antiviral interferon response, as well as production of interferon-stimulated genes. Protein kinase R, or PKR, after recognizing double-stranded RNA, leads to translational shutdown, as well as cell death pathways. And the OAS RNA cell system, after being activated by double-stranded RNA, leads to the cleavage of single-stranded RNA, both host and viral, as well as inflammatory responses and cell death pathways like PKR. Our previous work with MERS has shown that wild-type MERS does not induce these double-stranded RNA-induced pathways to any significant degree. Here's a Western blot for PKR activation, in which Senvis virus in the left lane is the positive control. You can see significant phosphorylation of PKR, as well as phosphorylation of eIF2 alpha, markers for PKR activation. When you compare this to the wild-type MERS-CoV lane, the third from the left, we don't see any phosphorylated PKR signal, nor any eIF2 alpha phosphorylation, at least above Mach levels. Similarly, here I show qPCR for interferon and interferon-stimulated gene expression, which has revealed that wild-type MERS does not induce interferon significantly. Even upon mutation of MERS innate immune antagonists, NS4A and NS4B, induction of interferon-stimulated genes in the interferon pathway, as well as an induction of PKR, is moderate at best. And this brings me to the central hypothesis of this project, that MERS shuts down double-stranded RNA-induced host pathways via the combined activities of NS4A, NS4B, and ENDO-U. NS4A is a double-stranded RNA binding protein, which sequesters double-stranded RNA from recognition by cellular PRRs. ENDO-U is an endoribonuclease domain within the conserved coronavirus non-structural protein 15 that has been shown to prevent accumulation of double-stranded RNA in other coronaviruses. And NS4B is a phosphodiesterase enzyme that functions as an RNA cell antagonist. You can see how these innate immune antagonists would limit activation of the double-stranded RNA-induced pathways. To further study immune evasion by MERS, we've used a panel of mutants that were generated in the FAIR and Paramount labs using a back-based recombination system. My data will highlight differences between wild-type MERS, its genome is shown at the top here, and various mutants, which I'll introduce quickly. A MERS mutant, the first of these is a MERS mutant expressing a catalytically inactivated NSP15, that is the non-structural protein with the ENDO-U domain. A MERS mutant deleted for NS4A, a double mutant that has mutated NSP15 as well as deleted NS4A, a catalytic inactivation mutation for NS4B, and another double mutant mutated in NSP15 as well as NS4B. Our work with these mutants has shown that significant induction of double-stranded RNA-induced host pathways only occurs when MERS accessory proteins NS4A or NS4B are mutated in combination with ENDO-U. Here's data on interferon production and A549s after infection with these mutants. As you can see, fold change induction of interferons lambda and beta are significantly higher in the MERS-CoV double mutants. Those are shown in this dark blue color and this cyan color over wild type and gray or any of these single mutants. And we see this pattern at early and late time points after infection. When we look at PKR activation using Western blot, we found that significant induction of the PKR pathway is only observed in the context of infection with the MERS, ENDO-U, NS4A double mutant. And similar to what I showed before, the activation of PKR is shown by phosphorylation of PKR, as well as phosphorylation of eIF2 alpha. And you can see that these readouts are absent or at mock levels in wild type MERS, as well as the single mutants in this panel of mutants that we tested. Finally, we used a bioanalyzer, which qualitatively assesses rRNA degradation as a readout for RNA cell activation. Both MERS double mutants, the NSP15 NS4B double mutant and NSP15 NS4A double mutant, activate RNase L, which is evidenced by this banded pattern in the lanes. Synvis here is a positive control, but RNase L activation in these double mutants still does not reach the level seen during Synvis infection. However, it is higher than what we see during wild type MERS infection, as well as various of these single mutants, which don't activate RNase L very much at all. Given the increased double-stranded RNA pathway activation during infection with these mutant viruses, we next pose the question, do MERS mutants with inactive ENDO-U produce higher levels of double-stranded RNA? Here I've shown representative immunofluorescence images in which we stained for double-stranded RNA with the J2 antibody. We consistently see increased double-stranded RNA production in MERS ENDO-U mutants, which corroborates data from studies in MHV. And we see this in the single mutant for NSP15, as well as the double mutants. And we went on to quantify this, where we find that double-stranded RNA signal is higher in the ENDO-U single mutant, as well as both of these double mutants compared to wild type MERS. Finally, when we quantified viral titers in these MERS mutants, we observed a reproducible log fold attenuation in both of the double mutants compared to wild type MERS and A549 cells, which are immune competent. When you compare that to these growth curves in Vero cells, there's no significant differences between wild type MERS and any of the mutants. However, you can see this significant decrease for both of our double mutants here. To determine which of these double-stranded RNA-induced pathways may be responsible for this attenuation in the MERS mutants, we quantified viral replication in A549 knockout cell lines that don't express either MAVs, which abrogates interferon signaling, PKR, or RNase L. So they're defective in they're each defective in one of these three pathways. While MAVs knockout, here you can see that in the wild type A549s, we see this reproducible attenuation in both double mutants. In the MAVs knockout cells, both double mutants are rescued. In PKR knockout cells, the 15-4A double mutant is rescued. And in the RNase L knockout cells, the 15-4B mutant is rescued, while the other double mutant isn't rescued in each of these. And so finally, to summarize the conclusions we've drawn from our work with these mutants, inactivation of ENDO-U results in increased double-stranded RNA production during MERS infection. MERS encodes at least 3 innate immune antagonists, NS4A, NS4B, and ENDO-U, which work together to shut down double-stranded RNA-induced host pathways. And our work with these pathways has shown that mutation of ENDO-U, along with NS4A or NS4B, is necessary for significant immune activation. Additionally, our work exploring our observed attenuation in these MERS mutants highlights the importance of these double-stranded RNA-induced pathways in restricting coronavirus replication. And that brings me to the conclusion of my talk or poster. Again, most of this work was done in collaboration with Courtney Komar. Here I've highlighted current members of the Weiss Lab, as well as collaborations from the FAIR Lab and Perlman Lab, who generated these MERS meetings, which allowed us to do this work. And thank you.